So to wrap up 7.1, I've got a couple of slides of ideas that I want to share with you here, and then I'm going to hand over for you to do your presentations and you to show me your thinking to finish off. So this slide here is all about non-coding DNA, and non-coding DNA has been referred to as junk DNA, and that's a misnomer. It's incorrect to call it junk, because actually this non-coding DNA is incredibly useful. And by non-coding, what we mean is that this particular section of DNA doesn't code for a polypeptide, but it does have a very important function. So this idea of language as a way of knowing is important. The first here is the regulatory roles of some parts of this non-coding DNA, and this will make far more sense after we've done 7.2. But what we'll do in 7.2 is we'll look at transcription, and in order for transcription to take place and produce something called messenger RNA, you need to have an enzyme bind to the DNA, the gene of the molecule, the DNA, and you have RNA polymerase, which is the enzyme that binds. And the RNA polymerase is going to bind at certain points before transcription can take place. And there are promoters, enhancers, and silencers which can interrupt that transcription process. We're going to come back to that later on. Then we've got this idea of satellite DNA, and satellite DNA are regions of DNA which seem to have very, very repeated tandem repeats, and we're going to look at how these variable number of tandem repeats, that's what this is, VNTR regions, are used for in gene profiling. We come back to this again also in Unit 4. There's also something called introns, and once again this will make far more sense after we've covered 7.2, but introns are part of the gene, so they interrupt the coding regions of the gene, and there's something called post-transcriptional modification where these introns are spliced out. But sometimes they can be spliced out in a different fashion, which means that even though you've got one gene, you can have variable versions of one gene. And this is a very interesting part of research which is going on in the world right now. Then we've also got sections of DNA, I call them genes here because they are referred to as genes, but be careful because normally genes code for polypeptides, but some parts of DNA, and I should put inverted commas here, they code for other things. For example, transfer RNA, and transfer RNA, ribosomal RNA, are very important molecules which are involved in protein synthesis, and there are sections of DNA that code for these. So all of these are non-coding DNA, that is that they don't code for a polypeptide, and in humans this makes up for the majority of our genome, which was revealed by the Human Genome Project. Good old BioNinja, they come up with a mnemonic, so I've added that there that could be helpful. And this slide here talks about how different species have got very, very variable numbers of coding and non-coding DNA. So this is the human here, and you can see that the amount of DNA which is responsible for coding polypeptides is really small. This red section here is the introns, and these introns are spliced out. We come to look at this in 7.2, and then we've got other non-coding regions just here. So this kind of data is useful to pause and take a look at, because this is the kind of thing that you might meet if you have a data analysis question. The next slide talks about introns and exons again. So this is a length of DNA, and this is a length of DNA which is what we're going to refer to as a gene. But in between, we've got sections which are called exons and sections which are called introns. And when we make the initial RNA molecule, we're going to do transcription, and we're going to make this copy here, which is going to be the same as the template, the same as the coding strand for the DNA here, but this is ribosomal RNA, so it's single-stranded, and what we've got here is a mixture of introns and exons. But then we need to appreciate that what happens post-transcriptional modification is we're going to splice out the introns. So again, this will make far more sense when we look at 7.2 and 7.3. Here, again, it's underscoring this idea of non-coding DNA that can be promoters, and I keep on saying it, but it is going to make much more sense when you look at the other Unit 7 high-level stuff. Here, this is where the telomeres are. So these are where the VNTR regions are going to be found predominantly. There's also in the centromere, this area here, there's also some variable tandem repeats, which are going to go here. So what I'd like to do is to hand over to you to present, and I've got slides which are in the PowerPoint, and within these slides there are all sorts of links and connections, so please dip into the PowerPoint as well, and we will complete this work on 7.1. Okay, it's quite complicated, and then what we're going to do is go back to the work on Unit 2, and we're going to look at transcription at standard level, and then bounce up to look at 7.2 and 7.3, which is the higher level version of transcription and translation.